Hello and welcome to this flow cytometric academic lab session. I am Badrinath. Today's talk is about CD34 positive hematopoietic stem cell enumeration methods. This assay being practiced in many of the clinical flow laboratories for more than 2 decades and has been well standardized and documented. In this talk, the focus is on 3 points. First, why do we enumerate stem cells? Secondly, I go through in detail the gating strategy used in a globally followed protocol called ISHAs. What is the importance of each histogram plot and gate used in this assay to define and report absolute counts of these stem cells? And finally, we talk a little bit about quality control and troubleshooting. Stem cells are CD34 positive, which can be isolated and harvested. Harvested product can be transfused into patients for engraftment of the bone marrow. Earlier transplant procedures used bone marrow progenitor cells as a source of stem cells. Now, the peripheral blood stem cell harvest rather than bone marrow is being used for autologous and increasingly allogeneic transplantations. Circulating stem cells are detectable in peripheral blood but are extremely rare. Can be increased and mobilized into peripheral blood with chemotherapy and or cytokines. Cord blood is also a source of these cells. In the context of transplantation, stem cell enumeration is important for clinical decision making. First, to determine enough CD34 positive cells have been collected. Count is critical parameter in engraftment. Generally, it is shown that for the successful engraftment in the recipient body, 2 million cells per kg body weight is considered sufficient. More concentration can yield quicker results. And secondly, it helps to monitor the rate of mobilization of these cells into peripheral blood with chemo and or growth factor administration so that clinicians can decide on the time of harvest of these cells. Harvesting can start with stem cells reaching 10 cells per microliters in mobilized blood. Less than 5 cells per microliter is considered above mobilization. Now we focus on methodology. Several guidelines for its measurement by flow cytometry have been proposed. Prominently, Milan protocol used a simple forward angle versus side angle light scatter to set a denominator. A positive isotope control window is used to establish CD34 positive region for CD34 stained cells. Bender is the first to introduce CD45 gating that works as a stable and more accurate denominator. We need a protocol which is globally accepted. Sutherland and others in 1996 developed a flow cytometry protocol called ISHAs that was to become a guideline. The idea of this protocol was to select the true CD34 events, minimizing interference from debris, dead cells which can bind antibodies non-specifically, by introducing a sequential Boolean gating strategy. And the good thing about this protocol is it can be used on a variety of flow cytometer platforms. This method has evolved over the time, a single platform method wherein absolute CD34 counts are generated on flow cytometry alone. This was introduced in 1998 by Kinney and others. ISHA's protocol has given specific recommendations on all aspects of the assay, starting from staining method, selection of antibody and fluorochrome combinations to be used, to reporting and data storage. Stain lyse no wash method is recommended. ISHA's method is a hierarchical system, uses CD34 and CD45 staining together with a sequential Boolean gating to identify true CD34 cells without any artifacts. Here, these cells are defined as they are CD34 positive, low SSE, low to intermediate CD45, and these stem cells are not smaller than lymph blast generic region cells. An absolute CD34 cell count is generated using a flow cytometry alone by incorporating counting beads. The inclusion of the viability dye 7-amino-actinomycin D will allow enumeration of viable CD34 positive cells. Based on ASHA's protocol, vendors have developed kits with automated ready-to-use reagents and softwares. For example, STEM kit from Beckman Coulter. In this, the counting beads are in liquid based, so they have to be added later into the tube. CD34 count kit by Deco, BD stem cell enumeration kit by BD Biosciences. In this, they have used true count tube wherein a known number of counting beads are pre-pipetted, so the pipetting errors are reduced. There are studies showing these kits essentially produce similar results. This is a general staining method to follow. Always process in duplicates. Ensure that WBC count is no greater than 40,000 cells per microliters. Use reverse pipetting. Isotype controls not needed. When adding liquid counting beads to the tubes, use same pipette which is used for the primary sample aspiration. Once stained, acquire immediately. Till then, keep in ice box. Now, we will go to instrument part. Once instrument is ready for startup, maintenance and QC is done, you can construct acquisition plots. Normally, for many of the flow assays, threshold is set for forward light scatter channel. In this plot, you see that the threshold is on fluorescent channel, that is FL1, where we have added CD45 antibody. This is because this assay uses stain lyse no wash method. Therefore, there will be a lot of lysed RBC debris. They have to be removed, otherwise they outnumber the cells of our interest. With appropriately set FL1 threshold, these debris can be excluded from the list mode data file. And also, counting beads are very small. If forward scatter is selected for thresholding, it will exclude acquisition of these beads. Therefore, a threshold is not set on forward light scatter. iShares recommends to acquire a minimum of 75,000 CD45 positive events with a minimum of 100 CD34 events to maintain a CV of 10%. The method is sensitive to at least level of 5 CD34 positive cells per microliter. Increasing the total number of CD34 events collected will allow for accuracy below this value. Now, I'll go through how analysis plots are created. I am showing these plots wherein we used a reagent kit called STEM kit by Beckman Coulter for staining. Samples acquired on Nevius instrument. As I said earlier, iShares protocol can be used on a variety of cytometers. What I mean is, these STEM kits can also be run in BD instrument, and BD STEM kit also can be used on Nevius. As a general approach, analysis is done manually by creating a series of 8 plots. First plot to identify viable cells, 45 versus SSC plot to select all leukocytes and a region to identify lymphocytes, a plot with a gate to include CD34 positive events and 3 serial plots to characterize two CD34 events. CD34 versus 45 plot to establish lower limit of CD45 expression by the CD34 events, and final plot to identify singlet counting bits to get number of bits collected. Now follow carefully how these plots with gates are created. In this 7 AAD versus SSC plot, include all 7 AAD negative viable events. These positive events which have taken up 7 AAD are dead, therefore these are to be excluded. And note that equus sample and commercial controls are stabilized products, that is, they are dead. So the viable cell gating region is open so as not to exclude any 7 AD stained cells. In a CD45 and SSC plot, display viable events, put a gate around all CD45 events, that is all leukocytes, while excluding CD45 negative events. Here you may use side scatter in log. In this log transformation, as you can see, small cell population is well separated. These strong CD45 positive cells are lymphocytes. Lymph region is set around CD45 mature lymphocytes. This gate will help to establish the minimum size range for the lymph blast generic region when they are shown in a forward scatter plot. This will be seeing in future plot. CD45 positive population is then displayed on CD34 versus SSC plot. Adjust the region to surround CD34 positive cells, including CD34 dim events. Note that this is a fresh sample, so you can clearly demarcate a cluster of CD34 positive events. If the sample is old or bone marrow or freeze-thawed samples, the cluster will not be this clear. A future plot coming in this sequence called guiding plot will guide you the limit of inclusion of this cell population. Hence, you may have to redraw this gate. The next is CD45 versus SSC histogram. Adjust the region to include cells forming a cluster with characteristic CD34 positive events with low side scatter and low to intermediate CD45 gating. Exclude CD45 brightly positive events, those are lymphocytes, and higher SSC events which are more mature mono or myeloid cells. This FSC versus SSC plot is to find size of the stem cells. Create two identical FSC SSC plots. In the first plot, display events of CD45 lymphocytes gated in a CD45 versus SSC plot. This region is adjusted to capture the cluster of lymphocytes. As mentioned earlier, when I created second plot, I said CD45 strong positive lymphos when showed in a forward scatter will define the size of lymphoblastic generic region. So we got the size of lymphocytes. Copy this gate on a second plot and link these two gates. This option is available for linking option is available for many of the softwares. In the second plot, capture CD34 versus side scatter cells. The linking of these gates is to ensure that both the gates are identical. Therefore, size of the cells are similar. This region identifies a cluster of events to meet the size criteria of ASH's guidelines for the CD34 stem cells. Events such as platelets and other non-specifically stained debris, if present, can be excluded. For the BT instruments, create one FSC SSC plot with an amorphous gate to include lymphocytes and duplicate this plot to include CD34 stem cells. The gates are automatically got linked. Now see whether our gating is proper by defining these 34 positive stem cells. That is, they are viable, positive for CD45 and 34, high CD45 and high SSC cells are omitted, platelets and or apoptotic bodies removed, and have the size of lymphocytes. This is what Aysh's sequential Boolean gating is about. In CD34 versus CD45 plot, display all events. Observe, this is ungated. This histogram is useful to visualize lower limit of CD45 expression within the CD34 positive events. This I said in the beginning, used as a guiding plot. Verify on histogram that the lower limit of region includes all CD34 positive events. Now region C is drawn to include all bead events. They are strongly positive, therefore may not be visible in this plot. This region must include the last channel of the FL1 log and FL2 log scale on the top. Time versus stem count. Display events from region C. These are singlet bead population taken as counting beads collected. In the Navier software, this region can be labeled as cal to allow automatic calculation of absolute number of CD34 positive cells. Type the correct acid concentration of the current lot of stem count fluorophores. Here they are 1012 beads per microliter as given in the kit insert. In BD Facts Canto 2, similar plots are drawn. See that the hierarchical gating is properly placed. When using BD's stem kit, you will notice the counting beads are readily visible. Now, to get absolute CD34 cells per microliters, use this formula. Events in CD34 gate divided by bead events collected multiplied by bead counts given in the kit insert divided by sample volume, that is going to be 100, and if we have diluted the sample, that should be multiplied with the dilution factor. When run in duplicates, the average CD34 count should be reported. This is a report page of Kaluza software, a third-party software. You can create similar reports on any Excel spreadsheet. Here is an example of results obtained. When run in duplicates, the results between two tubes should not vary more than 10%. You can also calculate percentage of CD34 cells and total WBC count. If CD3 is added in any other channel, fluorescent channel, you can get CD3 absolute counts and absolute MNCs and so on. It is a good practice to maintain a delta check in Excel sheet where you can observe the controls have passed and how the mobilization is progressing. WBC counts generated by flow, that is CD45 events divided by bead obtained and multiplied by bead concentration, and WBC generated by hematology analyzers when they match. That gives you an additional confidence that all aspects of your assay are right. Perform routine instrument setup with appropriate materials. Flow check for Beckman Coulter, CST beads for BD Canto. Color compensation settings Set as per your lab protocol, set to standard settings. As per internal and external quality controls are concerned, kit vendors supply controls to be set up once a day whenever CD34 testing is performed. Like STEC laboratories, CD checks comes with level 1, 2 and 3. BD stem cell control kit, CD34 high and CD34 low. Beckman Coulter's stemtrol has a known target concentration. However, we encounter problems with these kits these control kits such as first we have to order these controls separately as they are not part of staining kit. Shelf life of opened vial is about 10 to 15 days, and importing them on a timely basis in this part is a difficult task. In such a situation, it is, in my opinion, it is important to participate in proficiency testing programs provided by CAP, UK and RGPA and others. A survey was conducted in 2012 and later, I think 2018 by UK to see whether this protocol is properly followed. Surprisingly, this survey found many of the labs failed in its usage. The errors actually included omission of lymphocyte gate in CD45 positive leukocyte selection, omission of low SSC45 dim plot, selection of wrong parameter and wrong antibodies. Though they said it is ICS gating strategy, they have not used the gating properly. Non-use of any previously published gating strategies. Therefore, this survey addressed the issues of failure and tried to implement the protocol correctly by arranging workshops and advising on corrective steps. Finally, to be successful, follow proper gating strategy. Always use reverse pipetting. Do not change pipettes used in this assay. Dilute sample count should not go beyond 40,000 cells per microliter. If isoclonic controls used, subtract number of events from test sample events. And always participate in EQUAS programs. Thanks. I hope it will help you in your practice.